The body has many elegant mechanisms for eliminating potentially harmful compounds. Metabolic detoxification refers to the pathway by which the body processes and removes these harmful compounds. Metabolic detoxification is the process by which the body processes unwanted chemicals for elimination. The body metabolizes xenobiotics, or foreign chemicals, and unnecessary endobiotics, or endogenously produced chemicals, so that they can be excreted. Excess hormones, environmental toxicants, and prescription drugs are all cleared via the same enzymatic detoxification system. Metabolic detoxification is therefore essential for protecting the body from environmental factors and maintaining internal homeostasis. Natural interventions such as B vitamins and sulforaphane may help to support the detoxification pathways by promoting the activity of the essential enzymes. The body is generally very effective at detoxifying itself. Providing the body with essential vitamins, minerals, and other nutrients is the best way to ensure proper detoxification occurs. Many diets and trends that claim to detoxify the body are pushing the boundaries of scientific plausibility and may even be harmful. The metabolic detoxification process is comprised of 3 essential steps. Phase 1, enzymatic transformation. The purpose is to chemically transform compounds from lipid-soluble into more water-soluble. It's generally carried out by cytochrome P450 enzyme. Phase 2, enzymatic conjugation. The purpose is to further increase the water solubility and decrease the reactivity of the phase 1 products. It's generally carried out by UDP glucuronyl transferases, glutathione S transferases, and sulfotransferases. And phase 3 transport. The purpose is to excrete water-soluble compounds from the cells. It's generally carried out by ATP binding cassette transporters. Products of phase 1 are often more toxic than the original compounds. Phase 2 reactions neutralize the products to decrease their toxicity. Several factors, including dietary influence, smoking and consuming alcohol, advanced age, and certain diseases, may cause phase 2 enzymes to become overwhelmed, leading to toxicity, such as is seen in an acetaminophen overdose. Some steps you can do to minimize your exposure to toxins. You can choose cleaning products that are free of volatile organic compounds. Choose wood, tiles, or other alternatives for flooring instead of carpet. Use bisphenol A-free or phthalate-free plastic containers. Avoid warming food in plastic. Choose organic produce when possible. Wash fruits and vegetables before consuming and remove the peel. Limit the intake of processed foods. Cook foods at lower temperatures and avoid charring. Deficiencies in vitamin A, the B vitamins C and E are linked with decreased phase 1 activity, and it can slow the metabolism of certain drugs. B vitamins are also particularly important as cofactors in phase 2 reactions. Deficiencies in iron, calcium, copper, zinc, magnesium, and selenium have been shown to reduce phase 1 enzymatic activity. The reduced glutathione for glutathione conjugation requires adequate dietary sulfur-containing amino acids like methionine or cysteine for activity. Several flavonoids have demonstrated mild inhibition of multiple enzymes in animal models, and this can help when some phase 2 enzymes are overloaded. These include genistein, diadzin, and equal from soy, and theaflavins from black tea. Tannins from green tea can increase the cytochrome P450 enzyme activity in vivo, but also increase phase 2 activity. Sulforaphane, an isothiocyanate found in broccoli and other cruciferous vegetables, is among the most potent natural inducer of phase 2 detoxification enzymes. D-limonene has some chemoprotective activity due to its induction of phase 1 and phase 2 enzymes. In rats, D-limonene has been shown to increase the cytochrome P450 activity, intestinal activity, and liver glutathione activity. N-acetylcysteine can provide sulfur for glutathione production. It's effective at reducing oxidative stress, particularly due to heavy metal toxicity. The milk thistle-derived silymarin promotes detoxification by several complementary mechanisms. It acts as an antioxidant to lower the liver's oxidative stress that's associated with toxin metabolism, which conserves cellular glutathione levels. Artichoke and other plants and vegetables can stimulate bile flow, which is essential for toxin excretion. A variety of natural products have been shown in vitro or cell culture to directly increase the activity of phase 2 enzymes. These include the EGCG, or epigallocatechin gallate, from green tea, resveratrol, curcumin and its metabolite tetrahydrocurcumin, alpha-lipoic acid, alpha-tocopherol, lycopene, ginkgo biloba, allyl sulfides from garlic, among others. Other natural interventions that may be helpful for detoxification include calcium D-glucarate, chlorophyll, probiotics, and derivatives of quercetin. Detoxification has broad connotations ranging from spiritual to scientific, and it's been used to describe practices and protocols that embrace both complementary, such as fasting and colonic cleaning, and conventional, such as cleaning and antitoxin therapy, schools of medical thought, as well as some that push the boundaries of scientific plausibility, such as ionic foot detoxification. In the context of human biochemistry, detoxification can be described with much more precision. It refers to a specific metabolic pathway active throughout the human body, and it processes unwanted chemicals for elimination. This pathway, which will be referred to as metabolic detoxification, involves a series of enzymatic reactions that neutralize and solubilize toxins and transport them to secretory organs like the liver or the kidneys, so that they can be excreted easily from the body. This type of detoxification is sometimes called xenobiotic metabolism, because it's the primary mechanism for ridding the body of xenobiotics, or foreign chemicals. However, detoxification reactions are frequently used to prepare unneeded endobiotics, or endogenously produced chemicals, for excretion from the body also. Excess hormones, vitamins, inflammatory molecules, and signaling compounds, among others, are typically eliminated from the body by the same enzymatic detoxification systems that protect the body from environmental toxins, or clear prescription drugs from the circulation. Metabolic detoxification reactions, therefore, are not only important for protection from the environment, but they're central to the homeostatic balance within the body. There's some nutritional approaches for general optimization of metabolic detoxification. It's designed to provide a foundation for the proper functioning of the critical system. Specific health concerns may require supplementary detoxification intervention protocols, such as heavy metal detoxification or alcohol detoxification, reducing the risks. Toxins are poisonous compounds produced by living organisms. Sometimes the term biotoxin is used to emphasize the biological origin of the compound. Man-made chemical compounds with toxic potential are more properly called toxicants. Toxins and toxicants can exert their detrimental effects on health in a number of ways. Some broadly act as mutagens or carcinogens, causing DNA damage or mutations, which can lead to cancer. Others can disrupt specific metabolic pathways, which can lead to dysfunction of particular biological systems, such as the nervous system, liver, or the kidneys. The diet is a major source of toxin exposure. Toxins can find their way into the diet by several routes, notably contamination by microorganisms, man-made toxicants, including pesticides, residues from food processing, prescription drugs, and industrial waste, or, less frequently, contamination by toxins from other non-food plant sources. Some of the toxic heavy metals, like lead, mercury, cadmium, and chromium, while not man-made, have been released and redistributed into the environment at potentially dangerous levels by man, and they can find their way into the diet also. Microbial toxins secreted by bacteria and fungi can also be ingested along with contaminated or improperly prepared food. Even the method of food preparation has the potential for converting naturally occurring food constituents into toxins. For instance, high temperatures can convert nitrogen-containing compounds in meat and cereal products into the potent mutagens benzoprene and acrylamide, respectively. Smoked fish and cheese contains precursors to toxins called N-nitroso compounds, and these become mutagenic when metabolized by colonic bacteria. Outside of the diet, respiratory exposure to volatile organic compounds is a common risk which has been associated with several adverse health effects, including kidney damage, immunological problems, hormonal imbalances, blood disorders, and increased rates of asthma and bronchitis. One of the greatest sources of non-dietary toxicant exposure is the air in the home. Building materials such as floor and wall coverings, particle board, adhesives, and paint can off-gas, releasing several toxicants that can be detected in humans. For example, a toxic benzene derivative commonly used in disinfectants and deodorizers was detected in 98% of adults in the Environmental Protection Agency's study. In another EPA study, 3 additional toxic solvents were present in 100% of human tissue samples tested across America. Newly built or remodeled buildings can have substantial amounts of chemical off-gassing, giving rise to what's been called sick building syndrome. Carpet is an especially big offender, potentially releasing several neurotoxins. In testing of over 400 carpet samples, neurotoxins were present in more than 90% of the samples, quantitatively sufficient in some samples to cause death in mice. Ironically, short after the team report, 71 ill employees evacuated the new EPA headquarters in Washington, D.C., complaining of health problems which were eventually attributed to the 2,700 square foot of new carpet. Carpets also trap environmental toxins. The non-occupational pesticide exposure study found, on average, 12 pesticide residues per carpet sampled, and it determined that this route of exposure likely provides infants and toddlers with nearly all of their non-dietary exposure to the notorious pesticide DDT, aldrin, atrazine, and carbaryl. The driving force in the evolution of sophisticated metabolic detoxification systems was actually fairly straightforward and dependent on the ability of water to act as a solvent to dissolve substances. Since cellular membranes are primarily lipid-based and impermeable to most water-soluble, scientifically polar substances, the transport of water-soluble compounds into a cell requires specialized transport proteins. By placing the appropriate transport protein on the cell membrane, a cell will only allow desirable water-soluble molecules to enter, and it will prevent the entry of water-soluble toxins. This same paradigm also applies when the cell needs to excrete unwanted water-soluble compounds like cellular wastes. They exit the cell by a similar mechanism. In contrast to water-soluble compounds, the lipid cell membrane presents little barrier to lipid-soluble compounds, which can freely pass through it. Potentially damaging lipid-soluble toxins can then therefore gain access to cellular interiors, and they're much more difficult to remove. The metabolic detoxification systems address this problem by converting lipid-soluble toxins into inactive water-soluble metabolites. The solubilization of a toxin is accomplished by enzymes which attach or conjugate additional water-soluble molecules to the lipid-soluble toxin at specific attachment points. If the toxin doesn't contain any of these attachment points, they're first added by a separate set of enzymes which chemically transforms the toxins to induce these molecular handles. Following the solubilization reaction, the chemically modified toxin is then transported out of the cell and excreted. These 3 steps or phases of removing undesirable or harmful lipid-soluble compounds are performed by 3 sets of cellular protein or enzymes called phase 1, transformation, phase 2, conjugation, and phase 3, transport enzymes and proteins. Phase 1, 2, and 3 metabolism have different biochemical requirements and they respond to different metabolic signals, but they must work in unison for proper removal of unwanted xenobiotics, such as toxins or drugs, or endobiotics, such as excess hormones. Enzymes of the phase 1, 2, and 3 pathway have several characteristics that make them well suited for their important roles. Unlike most other enzymes, detoxification enzymes can react with many different compounds, broadening the number of toxins a single enzyme can metabolize. They're more concentrated in areas of the body that are most directly exposed to the environment, like the liver, the intestines, or the lungs. They're inducible, meaning that their synthesis can be increased in response to toxin exposure. The liver is the primary detoxification organ. It filters blood coming directly from the intestines and prepares toxins for excretion from the body. Significant amounts of detoxification also occur in the intestine, kidneys, lungs, and the brain, with phase 1, 2, and 3 reactions occurring throughout the rest of the body to a lesser degree. Phase 1 detoxification, enzymatic transformation. Under most circumstances, phase 1 enzymes begin the detoxification process by chemically transforming lipid-soluble compounds into water-soluble compounds in preparation for phase 2 detoxification. The bulk of the phase 1 transformation reaction is performed by a family of enzymes called the cytochrome P450s. Cytochrome P450 enzymes are relatively non-specific. Each has the potential to recognize and modify countless different toxins. After all, a mere 57 human cytochrome P450 enzymes must be able to detoxify any potential toxin that enters the body. However, the cost of this versatility is speed. The cytochrome P450 enzymes metabolize toxins very slowly compared to other enzymes. For instance, compare the predominant cytochrome P450 3A4 enzyme, which metabolizes 1 to 20 molecules per second, to superoxide dismutase enzyme, which metabolizes over 1 million molecules per second. Major sites of detoxification overcome the slower speed by producing large amounts of cytochrome P450 enzymes. It may represent up to 5% of total liver proteins, and similar large concentrations can be found in the intestines. The cytochrome P450 enzymes are among the most well-studied and best characterized detoxification proteins due to their role in metabolism of prescription drugs, and their role in metabolizing endogenous biochemicals, for example, aromatase, which transforms testosterone into estradiol. Several other enzymes contribute to the phase 1 process as well, notably the flavin monooxygenase. This is responsible for the detoxification of nicotine from cigarette smoke. Alcohol and aldehyde dehydrogenase, which metabolizes drinking alcohol, and monoamine oxidase, which breaks down serotonin, dopamine, and epinephrine in neurons, and its target for several antidepressant drugs. At a genetic level, the production of most phase 2 enzymes is controlled by a protein called nuclear factor erythroid-derived 2, or NRF2. It's a master regulator of antioxidant response. Under normal cellular conditions, NRF2 resides in the cytoplasm of the cell in an inactive state. However, the presence of oxidative stress triggered by metabolism of toxins from the cytochrome P450 enzyme activates the NRF2, allowing it to travel to the cell's nucleus. In the cell's nucleus, NRF2 turns on the genes of many antioxidant proteins, including the phase 2 enzymes. In this way, NRF2 senses oxidative stress or the presence of toxins in the cell, and it allows the cells to mount an appropriate response. NRF2 regulates the activity of genes that are involved in the synthesis and the activation of important detoxification molecules, including glutathione and superoxide dismutase. It also plays an important role in initiating heavy metal detoxification and the recycling of CoQ10, which is a potent antioxidant. Certain dietary constituents, including sulforaphane from broccoli and xanthohumol from hops, may also directly activate the NRF2 and stimulate antioxidant enzyme activity. This may partially explain their beneficial effects on detoxification. There are several families of phase 2 enzymes that differ significantly in their activities and their biochemistry. In several cases, phase 2 enzymes exhibit redundancy. A particular xenobiotic or endobiotic can be detoxified by more than one phase 2 enzyme. UDP glucuronyl transferase, or UGTs, catalyze glucuronidation reactions, the attachment of glucuronic acid to the toxin to render them less reactive and more water-soluble. There are several different UGTs that are distributed throughout the body, with the liver being the major location. In humans, many xenobiotics, environmental toxicants, and 40 to 70% of clinical drugs are metabolized by UGTs. The plasticizer bisphenol A and benzoprine from cooked meats are two notable examples of UGT substrates. A substrate is a molecule upon which an enzyme acts. Intestinal UGTs may affect oral bioavailability of several drugs and dietary supplements, and they may be responsible for chemoprevention in the tissue. Glutathione S-transferases, or GSTs, catalyze the transfer of glutathione, a significant cellular antioxidant, to phase 1 products. The GSTs play a major role in the metabolism of several endobiotics, including steroids, thyroid hormone, fat-soluble vitamins, bile acids, bilirubin, and prostaglandins. GST can also function as an antioxidant enzyme, detoxifying free radicals, and oxidized lipids, or DNA. GST are soluble enzymes that are ubiquitous in nature and in humans, forming about 4% of the soluble protein in the human liver, and they're present in several other tissues, including the brain, the heart, lungs, intestines, kidneys, pancreas, the lens, skeletal muscle, prostate, spleen, and the testes. Products of GST conjugation can be excreted via bile, or they can travel to the kidneys, where they're further processed and eliminated in the urine. Sulfonotransferases, or SULTSs, SULTs. They attach sulfates from a sulfur donor to endo or xenobiotic acceptor molecules. This reaction is important both in detoxification reactions as well as normal biosynthesis. The addition of sulfate to chondroitin and heparin, for example, is catalyzed by specific salts. Salts play a major role in drug and xenobiotic detoxification and the metabolism of several endogenous molecules, including steroids, thyroid and adrenal hormone, serotonin, retinol, ascorbate, and vitamin D. Salts in the placenta, uterus, and prostate are thought to play a role in the regulation of androgen levels. In contrast to other phase 2 enzymes, salts can convert a number of pro-carcinogens, such as heterocyclic amines from cooked meats, into highly reactive intermediates, which may act as chemical carcinogens and mutagens. While the UGTs, GSTs, and salts catalyze the bulk of human detoxification reactions, several other phase 2 enzymes contribute to the process to a lesser extent, but they're still important. Methyltransferase enzymes catalyze methylation reaction using the S-adenosyl L-methionine, or SAMe, as a substrate. Catechol O methyltransferase, or COMT, is a major pathway for eliminating excess catecholamine neurotransmitters like adrenaline or dopamine. Methylation reactions are one of the few phase 2 reactions that decrease water solubility. Arylamine N-acetyltransferases, or NATs, detoxify carcinogenic aromatic amines and heterocyclic amines. Amino acid conjugating enzymes, acryl-CoA synthetase and acryl-CoA amino acid N-acetyltransferase attach amino acids, most commonly glycine and glutamine, to xenobiotics. The food preservative benzoic acid is one example of a toxin that's metabolized by amino acid conjugation. Phase 3 transporters are present in many tissues, including the liver, intestines, kidney, and the brain, where they can provide a barrier against xenobiotic entry, or a mechanism for actively moving xenobiotics and endobiotics in and out of the cells. Since water-soluble compounds require specific transporters to move in and out of the cells, phase 3 transporters are necessary to excrete the newly formed phase 2 products out of the cell. Phase 3 transporters belong to a family of proteins called the ABC transporters for ATP binding cassette. Because they require chemical energy in the form of ATP to actively pump toxins through the cell membranes and out of the cell, they're sometimes called the multi-drug resistant proteins, the MRPs, because drug-resistant cancer cells can use them as a protection against chemotherapy drugs. In the liver, phase 3 transporters move glutathione, sulfate, and glucuronide conjugates out of the cell, into the bile for elimination. In the kidneys and the intestines, phase 3 transporters can remove xenobiotics from the blood for excretion from the body. The products of phase 1 metabolism are potentially more toxic than the original molecules, which doesn't normally represent a problem if the phase 2 enzymes are functioning at a rate rapid enough to neutralize the phase 1 products as they're formed. This, however, is not always the case. Factors which influence the ratio of phase 1 to phase 2 activity can upset this delicate balance, producing harmful metabolites faster than they can be detoxified, and increasing the risk of cellular damage. Some of the factors include diet. Some food and supplements can increase phase 1 enzyme activity. Smoking and alcohol consumption both induce phase 1. Age, which can decrease phase 2 UGT, GST, and SULT activity. Sex. Premenopausal women show 30 to 40% more phase 1 cytochrome 3A4 activity than men or postmenopausal women. Disease and genetics can also affect. An illustrative example of the consequences of phase 1 to phase 2 imbalance is the toxicity caused by overdose of the analgesic acetaminophen, also known as paracetamol, the active ingredient in Tylenol. Acetaminophen toxicity is the most common cause of liver failure in the United States. With a normal therapeutic dose of acetaminophen, the drug is predominantly detoxified by the phase 2 UGT and SULT enzymes. A small amount of the drug is detoxified by a third mechanism. It's first transformed into the toxic metabolite NAPQI by phase 1 cytochrome P450 enzymes, and this intermediate is then detoxified by conjugation with glutathione using the phase 2 enzyme GST. During acetaminophen overdose, the UGT and the SULT enzymes become quickly overwhelmed. Proportionally, more of the drug undergoes the third detoxification mechanism, transformation to NAPQI and conjugation by GST. Eventually, activity of the phase 2 GST enzyme slows as glutathione stores become depleted, and NAPQI is produced faster than it can be detoxified. The rising levels of the NAPQI in the liver cause widespread damage, including lipid peroxidation, inactivation of cellular proteins, and disruption of DNA metabolism. Treatment for acetaminophen overdose involves the timely replenishment of glutathione stores through the administration of the precursor amino acids for glutathione synthesis, most commonly N-acetylcysteine. Several other mechanisms work in concert with the phase 1, 2, and 3 enzyme systems to improve their efficiency or to extend their functionality. While not officially characterized as part of xenobiotic metabolism, they are nonetheless important for reducing or mitigating toxin exposure. Bile secretion is a critical digestive process for the absorption of dietary fats and fat-soluble nutrients, but it also functions as the major mechanism for moving conjugated toxins out of the liver and into the intestines where they can be eliminated. Antioxidation is a necessary protective measure against the harsh phase 1 oxidation reactions, which frequently produce free radical byproducts. The production of antioxidant enzymes, many of which are under the same genetic regulation by NRF2 as the phase 2 enzymes, is important for minimizing this free radical damage. Heavy metal toxicity can lead to oxidative damage by direct generation of free radical species and depletion of antioxidant reserves. Mercury, arsenic, and lead, for example, effectively inactivate the glutathione molecule, so it's unavailable as an antioxidant or as a substrate for xenobiotic detoxification. Lead can also reduce the activity of the enzymes that recycle glutathione. One method for heavy metal removal is their chelation by the cellular proteins metallothioneins, or MTs. These have a high capacity to bind various reactive metals like zinc, cadmium, mercury, copper, lead, nickel, cobalt, iron, gold, and silver. One molecule of MT can bind 7 to 9 zinc or cadmium ions, or any combination of these two, up to 12 copper ions and up to 18 mercury ions. Cellular stress, particularly oxidative stress, turns up the MT production, which, like phase 2 enzyme, is stimulated by the activity of NRF2. Prevention of absorption through trapping of potential toxins, such as surface adhesion to other molecules in the gut, like activated charcoal or kaolin clay, is an effective means of mitigating exposure. This mechanism has the requirement of some dietary adsorbents to be taken while the toxin is in transit in the gastrointestinal tract. Uptake of potential toxins and their detoxification by beneficial colonic microflora can also have a similar effect. Detoxification is the metabolic process of removing unwanted lipid-soluble compounds from the body. These unwanted compounds can be foreign, such as environmental toxicants, or endogenous toxins, such as excess hormones in nature. Detoxification reactions occur throughout the body, with the liver being the predominant detoxifying organ. Detoxification reactions follow 3 steps or phases. They have the ultimate goal of converting the toxin into an inert, water-soluble form for easy excretion. Phase 1 reactions transform the toxin into a chemical form that can be metabolized by the phase 2 enzymes. Phase 1 reactions are performed primarily by the cytochrome P450 enzymes. Phase 2 reactions, conjugates, attach the toxins to other water-soluble substances to increase their solubility. Each of the different type of phase 2 enzymes catalyze a different type of conjugation reaction. UDP, glucuronylyl transferase, or UGTs, catalyze the glucuronidation of most clinical drugs and several environmental toxins. Glutathione S transferase, or GSTs, conjugate toxins with the antioxidant glutathione. They can also directly detoxify free radicals. Sulfonotransferases, or SULTs, catalyze sulfonation reactions. They may also be important for controlling sex hormone levels. Other types of phase 2 reactions that are used less frequently include methylation and amino acid conjugation reactions. Phase 3 detoxification involves the transport of the transformed conjugated toxin into or out of the cells. Different phase 3 transport proteins work in concert to shuttle toxins from different parts of the body into the bile or urine for excretion. Following detoxification reactions, the toxins are removed from the body by excretion. Products of liver detoxification often leave the body by being secreted into the intestines in bile, but they can sometimes be transported into the bloodstream for processing by the kidneys. The cells that line the intestines can detoxify toxins as they're absorbed and release them back into the intestinal lumen. The kidneys can filter and further process toxins from circulation, excreting them from the body as urine. Given the sheer number of diverse enzymes and transport proteins involved in metabolic detoxification and its related pathways, it's no surprise that detoxification depends on and is sensitive to a large number of dietary factors. Macronutrients and micronutrient intake influences phase 1 and phase 2 systems. Protein deficiency decreases the cytochrome P450 metabolism, while high-protein diets increase it. The opposite effects are observed for carbohydrates. The effect of lipids on the cytochrome P450 metabolism is unclear. Efficient phase 1 reactions require sufficiency in several micronutrients. Deficiencies in vitamin A, B2, B3, folate, C, E, iron, calcium, copper, zinc, magnesium, selenium have all been shown to decrease the activities of one or more phase 1 enzyme, or to slow the transformation of specific drugs. The diverse set of phase 2 enzymes require an equally diverse set of essential nutrients, especially B vitamins as cofactors. The reduced glutathione for glutathione conjugation depends on adequate dietary sulfur-containing amino acids, methionine or cysteine. Vitamin B6 for the conversion of methionine to cysteine, as well as B2 and B3 for the activity of glutathione reductase, which recycles the oxidized glutathione. The metallation reaction uses SAMe as a substrate, which in turn is synthesized through folate and vitamin B12 dependent enzymatic reactions. The conjugation reaction of the NATs and amino acid acyltransferases uses the cofactor acetyl coenzyme A, or acetyl-CoA, which is synthesized from vitamin B5, using enzymes that themselves depend on multiple B vitamins. Several phase 2 reactions require the energy molecule ATP in some fashion. For example, the chemical cofactors for the phase 2 methylation, sulfonation, glucuronidation, and glutathione conjugation reactions are all made using ATP. These ATP-mediated reactions are all magnesium-dependent. Flavonoids have been extensively studied in vitro and in animal models for their ability to lower the activity of the cytochrome P450 enzymes and increase phase 2 enzyme activities, except for the salts, which they tend to inhibit. The inhibition of the cytochrome P450 activity by nerinogen, the principal flavonoid in grapefruit, has been well documented in humans. Hence, the recommendation to avoid grapefruit while taking prescription drugs. Other flavonoids that have demonstrated mild inhibition of multiple cytochrome P450s in animal models include genistin, diadzin, and equal from soy, and theaflavins from black tea. Green tea extracts and the quercetin derivatives isoquercetin and rutin are an exception to most other flavonoids. The green tea tannins can increase the cytochrome P450 activity in vivo, but it also increases phase 2 activity, the GST and UGT. Similarly, the quercetin derivatives were demonstrated to increase intestinal and liver cytochrome P450 enzymes in rats. Quercetin had no effect on the cytochrome P450s in this experiment. A wide variety of dietary components has been shown in vitro or cell culture to activate NRF2 and directly increase the activity of phase 2 enzymes. These include the epigallocatechin gallate, or the EGCG, from green tea, resveratrol, curcumin, and its metabolite tetrahydrocurcumin, which has the greater phase 2 activity, cinnamaldehyde, caffeic acid, phenethyl ester, alpha-lipoic acid, alpha-tocopherol, lycopene, apple polyphenols, ginkgo biloba, capsicum, and allyl sulfides from garlic. The beneficial effects of these phytochemicals have been demonstrated in numerous animal and human studies, particularly for their chemopreventative and antioxidant abilities. These effects may be explained by their indirect stimulation of antioxidant enzyme production and phase 2 detoxification through the NRF2 signaling. Isothiocyanates derived from glucoisonolates are reactive sulfur compounds with potent chemopreventative properties. Their prototypical member is sulforaphane, a constituent of broccoli that's the subject of several human cancer trials. Isothiocyanates such as sulforaphane and indoles such as indole-3 carbinol are among the most potent natural inducers of phase 2 detoxification enzymes. Sulforaphane and a derivative of I3C both directly activate NRF2, which increases the production of several protective enzymes, including GSTs, UGTs, glutamate cysteine ligase, which synthesizes glutathione, and NQO1. These derivatives are also strong inducers of many phase 1 and phase 2 enzymes, and thus are among the most well-studied phytochemicals for detoxification as well as for cancer prevention. Compounds from the Japanese horseradish, Wasabi japonica, and benzyl isothiocyanate from cruciferous vegetables similarly stimulate the phase 2 enzyme activity via the NRF2 activation. Both sulforaphane and the HITC also lower the cytochrome P450 activity. Sulfur constituents from garlic are inhibitors of various cytochrome P450s, and they induce GST and NQO1 activity in gastrointestinal tissue in rats. By activating the NRF2 components in garlic, they were able to reverse the depletion of antioxidant enzymes caused by toxic metal compounds in the liver of lab rats. D-limonene from citrus oil has been investigated for anti-cancer activity in uncontrolled human trials and in animal studies with some success. Part of the chemopreventative activity is due to the induction of the phase 1 and phase 2 enzymes. In rats, D-limonene has been shown to increase the total cytochrome P450 activity, intestinal UGT activity, and liver glutathione and UGT activity. Calcium D-glucarate is present in many fruits and vegetables, and it can be produced in small amounts in humans. When activated in the gut, it functions as an inhibitor of beta-glucuronidase, an enzyme produced by colonic bacteria and intestinal cells. In the intestines, beta-glucuronidase removes or deconjugates glucuronic acid from the neutralized toxins, essentially reversing the reaction catalyzed by the UGTs. Deconjugation reverts the toxin to its previous dangerous form and allows it to be reabsorbed. Elevated beta-glucuronidase activity has been associated with increased cancer. Chlorophyllin is a chlorophyll derivative, and it inhibits the cytochrome P450 activity, and it stimulates glutathione activity in cell culture and rodent models. The unique chemical structure of chlorophyllin and chlorophyll enable them both to bind and trap toxins in the gut, preventing their absorption. In animal models, chlorophyllin and chlorophyll lower the bioavailability, and they accelerate the excretion of several environmental carcinogens. Toxin trapping may partially explain the result of human trials of the results in Guizhou, China, an area with a high incidence of liver cancer due to the exposure to aflatoxin, a toxin produced by species of the fungus Aspergillus. Among the 180 people who took 100 milligrams of chlorophyllin 3 times per day, the urinary levels of the DNA aflatoxin conjugates, a marker for DNA mutation, went down 55% when compared to the untreated people. Certain strains of probiotic bacteria may minimize toxin exposure by trapping and metabolizing xenobiotics or heavy metals. Examples include the detoxification of aflatoxin and botulin, two toxins produced by Aspergillus, the metabolism of heterocyclic amines and dimethylhydrazine, and the binding of lead and cadmium. Additionally, the production of the short-chain fatty acid butyrate by lactic acid bacteria from the fermentation of dietary fiber has been shown to stimulate glutathione production in intestinal cell cultures. This may also contribute to some of the anti-carcinogenic properties of dietary fiber. N-acetylcysteine can provide an alternative source of sulfur for glutathione production. It's a free radical scavenger on its own, effective at reducing oxidative stress, particularly due to heavy metal toxicity. Because it can directly replenish glutathione stores, N-acetylcysteine is more effective than methionine at preventing liver damage, and it's the current treatment for acetaminophen or paracetamol toxicity. It's an effective treatment for acute liver failure due to non-acetaminophen drug toxicity also. Milk thistle, the most well-researched plant in the treatment of liver disease, contains a mixture of several related polyphenic compounds called silymarin. Silymarin promotes detoxification by several complementary mechanisms. The antioxidant capacity of silymarin can lower the liver's oxidative stress associated with toxin metabolism, particularly lipid peroxidation, which has the effect of conserving cellular glutathione levels. Like N-acetylcysteine, silymarin can protect against acetaminophen toxicity, possibly by the similar mechanism of preserving glutathione levels. Silymarin, however, may be more effective antidote than NAC for acetaminophen toxicity if the treatment is delayed. In animal models, it was effective when administered up to 24 hours after an overdose. Phase 3 transporters, while important for removing toxins from healthy cells, can also decrease the effectiveness of pharmaceutical therapies by increasing their clearance. This can be especially problematic with chemotherapy drugs, which phase 3 transporters enable cancer cells to become more resistant. Therefore, stimulation of phase 3 activity may not always be desirable. Dietary factors have differing effects on phase 3 transporters. For example, apple polyphenols and sulforaphane, at least equivalent to about 2 servings of broccoli, both stimulate the activity of the phase 3 proteins. In contrast, the curcumin metabolite, tetrahydrocurcumin, decreases the activity of the phase 3 transporters in human cervical carcinoma and breast cancer cell lines. Resveratrol decreases phase 3 protein synthesis, which prevented acute myeloid leukemia cells from becoming resistant to the chemotherapy drug doxorubicin in cell culture. Silibinin, the chief constituent of milk thistle, is also a phase 3 inhibitor, both in vitro and in vivo. As a major carrier of toxins from the body, proper bile flow is a critical final step in the metabolic detoxification process. Impairment of bile flow, cholestasis, resulting from dysfunction within the liver or blockage of the bile duct, can result in the buildup of liver toxins and liver injury. Cholestasis can also be the result of the detoxification process itself. There is increasing evidence that the detoxification and excretion of clinical drugs into the bile can produce cholestatic liver disease. Artichoke has been used for centuries in folk medicine as a liver protectant and to stimulate bile flow, and is best studied for a choleretic agent. Artichoke contains several antioxidants that can protect against oxidative liver damage, as well as caffequinoic acids, which have been shown to stimulate bile flow in animal models. Caffequinoic acids may also be responsible for the choleretic properties of yarrow, fennel, and dandelion. Androphyllus, garlic, cumin, ginger, and carom seeds, as well as mustard and curry leaf, have also been shown to stimulate bile flow or bile acid production in rodent models.